Yes, so antibodies are actually used a whole lot in research, but they're also used in other things as well. But when you guys think of antibodies, what do you initially think of? Sickness, blood, vaccines. Yeah, so these are all things to do with the immune system, right? So antibodies are actually, antibodies use in research is something that we've kind of jumped onto after seeing what antibodies do in the body. So it's not something that was just generated in a lab, but rather we saw the usefulness of antibodies in terms of the immune system, and then we're able to harness what they can do to apply to different things like research and therapeutics and diagnostics. So what is the general shape of an antibody? Yeah, so you would describe it as a Y, right? Yeah. Just be happy. I had this activity that I was going to make you do, which involved you all standing around being antibodies and picking balloons up off the floor, but vetoed that idea, so you don't have to do that. But yeah, so antibodies are also known as immunoglobulins. And so you'll see this immuno come up a lot in a lot of the things that I'll be talking about today because, like we said, it's associated with the immune system. So antibodies are these large Y-shaped proteins, and they're produced by specialized cells called plasma cells, and we'll talk a little bit more about that in a minute. And they have a role in identifying and neutralizing pathogens. I'll scooch this way. So you can see the general shape of an antibody is this Y shape, and then, so this is just kind of a schematic, and then here you have a ribbon structure, and it still kind of follows this Y shape. There are different types of antibodies, and you'll notice down here that these are distinctly not Y-shaped when you're just initially looking at them, but then break it down. So your IgM is actually a pentameter of these Y-shaped antibodies, and then your IgA is two that are joined end to end. So the Y shape is the initial, like the antibody shape. So when you're looking at this, antibodies have, I keep forgetting I can't see it on my computer. The V is basically where you're going to be binding to your antigen. So these are antigen binding domains, and these are constant regions. And that's not very important in terms of what we'll be talking about today. So are any of you taking immunology right now, or have you taken immunology? Yeah, so it's really, really complicated, and I am in no way going to get into the details of antibody specificity or how all of this comes about. I just kind of want you guys to know the basics of antibodies being derived from the immune system, because it'll help explain some of the specificity and the uses of them later on. But because they come from the immune system, I'm just going to give you a quick rundown of the immune system. So you guys remember that you have two arms of the immune system, the adaptive and the innate. Does anyone want to do a quick rundown of what is the innate immune system? Like if you had to sum it up in a sentence.
It's the basic barriers and checkpoints and like things that will generalistically respond to something getting into your system, but they'll respond the same way every time essentially to the same threat.
Right, exactly, yeah. So a lot of times we think of the innate immune system as your first line of defense. So it's just what you have, initially. So if you're exposed to some kind of pathogen, your innate immune system is going to be able to respond to that. And it's generally an immediate response, right? So you don't have this lag in response. It's something that's going to happen right away. And while it has this immediate maximal response, there's limited specificity. So your innate immune system is just kind of recognizing, I guess you would call like broad generalized patterns that can be seen in different microbes or pathogens. And so these things can include things like barriers. So your skin is a barrier, tears, flushing things out of your eyes, and then different types of cells, including phagocytes, which include neutrophils and macrophages, natural killer cells and mast cells, and then complements and other protein systems that kind of help to neutralize and bind to these pathogens. So one thing I didn't list there that's important is the dendritic cell. And why might the dendritic cell be important in terms of innate and then going into adaptive immunity? Any guesses? Okay, so dendritic cells are antigen-presenting cells. So they will engulf these pathogens and then present these antigens on the surface where they can be exposed to T lymphocytes. So basically, to put it simply, dendritic cells kind of serve as the linking point between the adaptive and the innate immune system. So your dendritic cells are exposing your adaptive immune system to what's being seen. And once the adaptive system sees this, this antigen, it can suddenly start its response. So the adaptive immune system is your second line of defense, and the response is slower because it has to be triggered by an antigen recognition, like with the dendritic cell presenting what it sees during that innate first line of defense. The difference between the two, aside from what we've already mentioned, is that the adaptive immune system is very, very specific, and it has what we call immunological memory. So we generally break down the adaptive immune system into two categories. We have humoral and cell-mediated, and humoral just kind of refers to humors, body fluids. It's like the old term for that word. And the humoral arm of the adaptive immune system includes B cells, those plasma cells that we talked about earlier, and these are responsible for secreting antibodies into blood and extracellular fluid. The cell-mediated arm of the adaptive immune system includes cytotoxic and helper T cells. So those would be the CD8 and CD4 T cells that you hear about. And these kind of help to support antigen recognition and then destruction of infected cells. So where do we commonly hear CD4 T cells referred to? Like, it's something that you hear a lot in terms of specific disease. Sorry, what was that? So cancer, and then I'm thinking more in terms of like immunosuppressed people, diseases, HIV, right? So when we're talking about HIV, a lot of times you'll hear about CD4 T cell counts and things like that. And we know that HIV is an immunodeficiency virus, right? So the virus actually preferentially targets and infects these T lymphocytes. And in doing so can wipe out your immune system. And that's why when you develop AIDS, you're susceptible to a whole host of other diseases that you would normally be able to fend off because your immune system is functioning. And after you've progressed to AIDS, you're not really functional anymore. So this is just like a little comic that we'll walk through to make sure you guys get this idea. So here you have your happy little B cell. And these are incoming antigens that are being presented on bacteria. And so once the bees are, I can't see very well. So once the B cell has been activated by being, by responding to this antigen that's been presented to it, it can go and initiate activation of this helper T cell, which then helps to initiate clonal expansion. And we'll talk about that in the next slide. But basically what this plasma cell is going to be doing from here on out is just shooting out these antibodies. So it's making more and more of these antibodies, and these antibodies are then able to attract other immune cells after they kind of latch on to their target. And so when I say that there's immunological memory, what I mean is that once these B cells have kind of differentiated and recognized their antigen, they make more and more copies of themselves, and they also make these memory cells that persist in the body, so that the next time you see this antigen, which would be like the next time you see this pathogen, you already have a response waiting and ready. Does that make sense? Okay, so I mentioned this idea of clonal expansion or clonal selection. And basically what's happening is that you have these lymphocytes that are being generated, and each lymphocyte progenitor gives rise to a whole bunch of different lymphocytes. And these lymphocytes all have unique receptors for antigens. You guys following me so far? So each individual cell can recognize a specific antigen. And so you have a whole bunch of different lymphocytes running around with specific receptors for specific antigens, but they're not all making little armies of themselves. They have to be activated first. And another thing we have to worry about is what if these lymphocytes can recognize your own body's antigens? Like when would that be a problem, do you think?
When you have the antibodies recognize self?
Yeah. And what kind of problem would that be? Like what would that lead to? Right, yeah. So a lot of these autoimmune issues that we see are arising from your body recognizing self as non-self. So it's attacking self-antigens, which is a problem, right? So generally speaking, the body's pretty good about eliminating these cells that have self-antigen recognizing receptors so that you don't have this autoimmune problem. So once these antigens are interacting with these lymphocytes, the cell is then activated, and when the cell is activated, it gives rise to kind of like a clonal army. So you have all of these antibodies waiting and ready. And so remember, this is all happening here at these, it does match up. It wasn't for a while. You have these variable regions, which are actually the parts of the antibody that are recognizing the antigen. And what's really cool and what was a mystery for a long time is how you could have so many different receptors for all of the possible antigens out there encoded in your cell line. And that's another super complicated thing, but basically you have this cool process of somatic recombination that occurs in the germline of these cells so that you get this wide range of potential antigen recognizing domains, which if you take immunology, and you should because it's really cool, you'll learn more about. It's a really fascinating process, and it was seriously a huge mystery for a really long time. Like, how can we have so many different possible recognition receptors for all of these potential antigens that are out there? So one thing that's really important to come out of all of this is this idea of immunological memory. And so you see here, this is just kind of a timeline and antibody concentration. So when you're first exposed to an antigen, you form antibodies to it over time, and then those will kind of drop down as you no longer need them. But then once you're exposed for a second time, look at what happens to your antibody response. So this is where you have that clonal expansion background. You've got everything waiting to respond. And so where have we taken advantage of this response and used it as a tool? Yeah, so basically when you're vaccinating or immunizing, you're taking advantage of immunological memory. So generally when you're vaccinating or being vaccinated, you're exposing a person to some sort of antigen, and it might not necessarily be the entire pathogen. So you can have just the antigen from a specific virus rather than the entire virus itself. So you're not actually putting a person at risk of developing the disease, but you're priming the immune system to recognize that antigen so that the next time it sees it, which might be when it's actually the entire virus, your body's ready to fight it off. So that's kind of the background on how vaccinations came about. And so we've been talking about antigens and antibodies, and are you guys clear on what an antigen is compared to an antibody? Okay, so because those are a little tricky sometimes to keep straight. So the antigen is whatever foreign particle that is eliciting the immune response. So whatever it is, it can be a protein, sometimes it's a lipid, it can be a number of things, but whatever is eliciting the immune response is the antigen. And the antibody is what's being generated to fight it off, I guess, is how I've always thought about it. But again, it's those proteins that are being produced by those plasma cells. And they recognize specific epitopes. And that's really important that it's specific on antigens. And this facilitates the elimination. So what do I mean by epitope? Yeah, so we say antigens, right? But antigens can be kind of bigger, I guess you can think of it. Epitopes are these specific little areas on something that are being recognized. So antigens can have several different epitopes. And paratopes are kind of the receiving end of the antibody. So there's a lot of specificity, and a lot of times it's kind of compared to the lock and key analogy of how things fit together, that you're only going to have one key for one lock. So you're going to have one antibody for one antigen. That being said, there are instances of cross-reactivity, but to make things simple, just think of it as one antibody for every antigen. And And the epitope matches up with the paratope. So the epitope of the antigen matches up with the paratope of the antibody. Does that make sense? Okay. So how, like, why would this specificity be useful, do you think? Like, can you think of any applications for such specificity?
You could use it to identify, if you generated those antibodies, you could use them to identify a substance that you're trying to find in a sample.
Yeah, exactly. Because it's so specific, it's really useful if you're looking for something specific. And due to their specificity, we use antibodies for a lot of things. And I've already mentioned their role in therapeutics, diagnostics, and research. And so actually the market for antibodies is huge. Some of the stats, like, I don't know if you can see the numbers here, but the money that's coming in from antibodies at 30 billion in therapeutics, 28 billion in research, and what is that, 88 billion in diagnostics. So antibodies are kind of a hot commodity right now. And so for a long time, it was just antibodies that were being generated in labs, but now they're also doing chimeric antibodies, and they're doing non-traditional antibodies. There's a lot of new technology that's going into developing antibodies as therapeutics. And so as therapeutics, they can be used to treat autoimmune disorders, inflammatory diseases, and cancers. And then for diagnostics, you see them in pregnancy tests and disease detection assays. So your little pea on a stick pregnancy test is basically just an ELISA on a stick. And we'll talk a little bit about ELISAs in a minute. But then again, we also use a lot of antibodies in research. Immunoprecipitation, flow cytometry, immunocytochemistry, immunohistochemistry. Have any of you used antibodies in your research? You have? You will be. Yeah, so I'll talk a little bit about therapeutics and diagnostics, and by talk I mean I have some videos you guys can watch, so I can stop talking for a minute. And then we'll talk about how they're being used in research, because that's kind of where you guys are more likely to come across them when you guys are working in the labs. Okay, so I guess the first question though is, where are these antibodies coming from that we're using for therapeutics and diagnostics? Like, where do you think they're coming from? You just go out and buy antibodies? Like, how do you get antibodies? Guesses? I'm sorry? Yeah. So you can just go out and buy antibodies, but they have to be created first. They have to be generated. Like, I just bought a whole bunch of antibodies last week, and I love that you can do that. And just go online and find the ones you need and order them. But somebody somewhere has to generate the ones that you're going to be using. And so that brings me to this idea of polyclonal versus monoclonal antibodies. When you guys are going out and ordering your antibodies, you'll notice that there is a huge price difference in polyclonal versus monoclonal, and that is due to how they're generated. So polyclonal antibodies are kind of, so they're all against a specific antigen, but they might match different epitopes of that antigen. Does that make sense? So we talked about how one antigen can have different epitopes. So these polyclonal antibodies will recognize different epitopes, and they arise from multiple B cell lineages. So you have all of these different plasma cells that recognize a different epitope on an antigen and then generate antibodies for those different epitopes. Whereas monoclonal are coming from a single B cell lineage. So they're a bit more specific, and they're always going to target the same epitope on an antigen. So if you're worried about like specificity or being able to very neatly reproduce something in the lab, monoclonal is the way to go. But the process of making monoclonal antibodies is a bit more complicated. So when you're generating polyclonal antibodies, and I don't know if you knew this, a lot of times we generate antibodies in animals. So you'll see a lot of times you'll see primary antibodies that are like mouse, anti, whatever your antigen is. So say I'm working with cholera toxin, which is an antigen that is found in cholera, like the surface of cholera. So you can generate antibodies against cholera toxin. So you can buy mouse anti-cholera toxin, goat anti-cholera toxin, donkey anti-cholera toxin, and that just refers to whichever animal the antibody was raised in. So when you're doing polyclonal, you're just injecting an antigen into an animal. And I mean, this is very simple. I'm sure they don't look that happy when it's happening. But you have a little animal, you're injecting antigen into the animal. The animal's lymphocytes are producing antibodies in response to the antigen. You remove a sample from the animal, and then you spin down to isolate antibodies. Does that make sense how that works? Yeah, so pretty straightforward. You don't need anything particularly magical to be able to do that. Versus monoclonal antibodies, remember you're wanting to have this from a single B cell lineage. So you only have a specific epitope on a specific antigen. And in doing so, you're immunizing an animal with your antigen of choice. And then you are screening for these B cells that are producing these antibodies. And these B cells are then fused to tumor cells that are constantly regenerating to form these hybridomas. These hybridomas then secrete the antibodies, and then the antibodies are cloned, or the cells are cloned, and then you harvest your antibodies. So it's a bit more time-intensive, money-intensive, which is why the monoclonal are more expensive when you're buying them. But like I said, it gives you some advantages in the lab when you're trying to look for something very specific. All right, so, oops, let's see how I can make this work. Oh, let's see. Do you know, can I switch to this computer for a minute? And then I have to figure out how to find my presentation again because I just lost it. Let me do this for a second. Cool. Thanks.
I'm going to go back, just hit that one.
Does anyone know, can you embed videos into PowerPoint from YouTube?
Like the whole video?
Yeah.
I'm not sure the volume's not ridiculous.
I'm not saying like I want to do it right now, just for future reference.
Oh.
Okay. So, I don't, fine. Apparently we're signed into John's. Okay, that's a commercial. So this is, pause this for a second. So we talked a little bit about applications of antibodies in therapeutics and diagnostics, and I thought it would just be a lot easier for you to see how antibody treatment works for a specific therapeutic target, so in this case cancer, than me trying to explain it to you. So let me turn this down a little bit, and hopefully this makes sense, and I can stop talking for a minute.
Cancer is a complex disease, and responses to existing therapies are often insufficient or short-lived. The key challenge is that tumors are heterogeneous structures containing diverse cancer cell populations that interact with an intricate network of surrounding stromal cells that support tumor growth. Cells communicate with each other by molecular signaling. Typically, in cancer cells, growth signals come from receptors on their cell surface. A growth signaling event is often mediated by the binding of a growth factor, or so-called ligand, to its matching receptor. Ligands may be produced by the tumor cells or the surrounding tumor stroma. For many years, single monoclonal antibodies have been used to treat cancer by preventing the ligand-receptor interaction. Some target the receptor, like Herceptin, and some block the ligand, like Avastin. However, tumor resistance to such therapy develops rapidly. This is often due to the interaction of the ligand with its receptor being incompletely blocked by a monoclonal antibody, allowing the cancer cells to survive treatment. Or, the cancer cells adapt and continue to grow by using alternate receptors that mediate similar growth signaling. In addition, it is common within a tumor to find cell populations that grow independently of the block receptor, perhaps through other receptor-ligand interactions, and they consequently evade monoclonal antibody treatment. Researchers at Synthogen have developed an innovative approach to conventional monoclonal antibody therapies that makes it feasible to simultaneously target multiple binding sites or multiple receptors with a single antibody mixture. A mixture of antibodies binding distinct sites on the same receptor not only blocks the ligand-receptor interaction, it can also more effectively cross-link the receptors than a single monoclonal antibody, causing rapid removal of the receptor from the cell surface. An antibody mixture applied against multiple receptors simultaneously effectively reduces a tumor cell's ability to find alternate growth signaling pathways. With an antibody mixture, it is possible to launch a very specific and yet multi-targeted attack with just a single administration. This offers the unique opportunity to simultaneously target tumor cells and surrounding tumor supporting cells, and to prevent tumor resistance associated with tumor heterogeneity and adaptability.
Okay, does that make sense? You see how antibodies can be very useful when you have a specific target in mind. Crap, so now I have to switch back. Sorry guys, the hazards of trying to do a Mac and a PC. Let's see if I can make it work like it did. Oh. Okay, this is weird. So what I see up here is not what I see on my computer screen. So I had another video for you, but I think I'll wait and we'll watch that in a little bit because you guys are going to do an activity that relates more to the video. But as far as research applications, I'll just review some where we commonly use antibodies in the lab. And so Western blots are one that we very frequently see in papers and people talk about. Have any of you done a Western blot? No? Okay. Wait, you've done a Western blot. It's like, I know someone in here has. Thorne's done a Western blot. So Western blots are is a technique that we use to look for proteins in a sample. So I guess I'll have a picture of it, but basically, remember to bring this with me. If you've seen a figure in a paper where you have like a blot and then a bunch of bands, usually they're black and white. You have little molecular weight markers here. So this is something like a Western blot. So generally speaking, when you're running a Western blot, you take a sample with your proteins in it and you run it through a gel, and it's not like an agarose gel, it's a polyacrylamide gel. And this separates out your proteins based on weight. And you can't see them, right? They're just in this gel. You then transfer this gel onto nitrocellulose or some other sort of membrane. So sometimes PVDF is what we use. And that transfers the proteins onto the membrane. Once the proteins are on that membrane, you want to visualize them. And you can find these proteins by using antibodies that are specific to the protein that you're interested in. So you then incubate this membrane with your specific antibody, and you're able to detect. So there are different detection methods that are associated with Western blots. And those can include colorimetric detection, chemiluminescence, or this fluorescence. And so colorimetric Western blots will give you some sort of color change associated with your antibody. So what generally ends up happening is you have an antibody that is either directly linked to some sort of enzyme, or you have an antibody that then is recognized by what's called a secondary antibody that's linked to an enzyme. And then when you add a substrate, there's a color change. Does that make sense? It gets a little bit complicated. So when you're thinking about enzymes, you can either have, or enzymes, antibodies, like say this is your target antigen. You can have an antibody that binds directly to that and then has something that is going to allow you to visualize it attached to it, or you'll have an antibody that binds to that, and then something that binds to that antibody that has some sort of marker for detection on it. Does that make a little more sense? Okay, so this would be your primary antibody. This would be your secondary antibody. So when you hear people talking about the primary antibody species they used or the secondary antibody, it's just what order you're using it to detect your antigen of interest. So you can have a colorimetric detector. So that would mean that whatever this little marker is going to give you a color change, and that's how you're going to see your proteins. Or you can have a chemiluminescent one, which is kind of cool. So I used to do these ones all the time. But you have your membrane and you have the substrate that you add to it, instead of making a color, will actually give off light. So a lot of times we develop these on film in a dark room. And so it was great because I'm clumsy. Sometimes I would drop my membrane, but these things glow in the dark. So you can like see your proteins glowing on your membrane and find wherever you dropped your membrane. And then you also have these fluorescence ones. And I've never actually done a fluorescence Western blot, and I didn't even know these things existed until today. But these are, yeah, these are pretty cool because they actually allow you to detect multiple bands or multiple samples within a membrane. You don't have to incubate with different antibodies and then develop that antibody, go back and strip it off, and then develop the next one. With the fluorescence antibodies, you can be very specific and do multiple ones at the same time because they'll fluoresce at different wavelengths, so you get to see them separately. All right, so Western blots, we use lots of antibodies. Also in immunohistochemistry and immunofluorescence. So if you ever see these really beautiful pictures of glowing cells, it's very likely that antibodies were involved in detecting whatever is glowing. Has anyone done any histochemistry or cytochemistry that involves fluorescence? No? Okay. So histochemistry, like you could almost classify immunofluorescence as a type of histochemistry. So histochemistry is just kind of looking at tissues versus cytochemistry, which is looking at cells. And then immunofluorescence is just attaching a fluorescent probe to any of these immunoprobes that you're using to visualize. So this first image here, you have, I don't remember what it is, just a bunch of cells. So it's a tissue sample that's been stained using immunofluorescence, or not immunofluorescence, detection. So you get a color change, but you're not really getting that glowing. With immunofluorescence, it's really great because you can incubate cells or whatever you're interested in with specific antibodies that will glow at specific wavelengths. So this series of images here are all the same cells. They're just with antibodies that are directed at different things. So this green one is tubulin. No, green is actin. The red is tubulin. And then the blue is a stain for DAPI. And so that targets DNA in the nucleus. So you can see all of these different structures in one sample using immunofluorescence because they're all so specific. The antibodies that are being used are only going to react with whatever the antigen that they're specific for is. So you can get these great overlaying images where you see all of them together here. So we can also use antibodies in immunoprecipitation and flow cytometry. So same general idea where you have an antibody that's binding to its specific antigen, and in the case of immunoprecipitation, you can make sure that the antigen-antibody complex is insoluble, so you can pull it down and spin it out from the rest of a sample. And that helps if you're trying to, purify a whole bunch of protein from one specific sample or see what's interacting with those proteins. And it gives you a way to be very specific and targeted in pulling that out. Flow cytometry is a method that lets you sort and count cells. So you tag your cells with fluorescent antibodies and run them through a counter, and you can see how many of a specific cell type you have, or how many of a cell that expresses some sort of receptor that you're interested in. And then you can also use this type of method to sort them. So if you're trying to sort out cells in a population. All right, so mentioned ELISA a couple of times. Have any of you done an ELISA, either in the lab or in a class? Okay, yeah, so I can't remember if it's cell or micro, but one of the two bio classes, I know you do ELISAs in the labs. But ELISAs are, I guess, ELISAs are a technique that we use a lot in the lab, and they're really pretty easy and they're relatively cheap for what you're trying to do. But it's this idea, again, of using antibodies to recognize an antigen. And you can do this in a sample to see if, for example, a person is expressing HIV antibodies because they've been exposed to HIV and they have the virus. So ELISAs are used a lot of times for diagnostic tests, but we also use them in the lab. Like I was using them to determine concentrations of specific proteins in response to some stressor, because you can quantify based on how much you have in your wells. So let me see if I can get this video working. Crap, that's not the right one. Watch.
The enzyme-linked immunosorbent assay, or ELISA, is an extremely sensitive test that is used to detect antibodies or specific antigens. The direct ELISA is a test for the presence of antigen. In this procedure, a known antibody is adsorbed to the inside of the well in a microtiter plate. After rinsing to remove excess antibody, the sample suspected of containing the antigen is added. Next, an enzyme-linked antibody that can react with the antigen is added. If antigen is present in the well, the enzyme-linked antibody binds to it and is retained. The colorless substrate for the enzyme is then added. Development of color indicates the presence of the antigen. In the indirect ELISA, antigen is added to the microtiter plate well, and the antigen attaches to the walls of the microtiter plate. After rinsing to remove excess antigen, the serum suspected of containing the antibodies is added. Enzyme-linked antibody capable of reacting with the constant region of other antibodies is then added, followed by addition of the colorless substrate. Development of color indicates the presence of the antibody being identified.
Okay, does that make sense? Do you see the use of two antibodies there? Kind of like with the Western blot, the primary and the secondary? Okay, good.